Hello everyone and welcome to my channel Biology Made Easy for Exams and Interviews. Today we are going to talk about 3-point cross. Now 3-point cross in genetics is very important to understand the very basic concept of chromosomes, the concept of genes on chromosomes, the distance between the genes. So before that, let me give you a very brief overview that we now know that a chromosome contains double-stranded DNA. And we also know that there are genes on the chromosomes, and these genes are in pairs. That means this is N and it is in diploid 2N, right? And we also know a pair can be homozygous or it can be heterozygous. So let's say this is heterozygous condition, such that capital A is the dominant on small a. So this is a dominant, this is recessive. We know that, right? In Mendelian genetics, when Mendel discovered certain traits or characters, right? Mendel said traits or characters which are present on certain locus or loci. He said that, okay, and he gave the concept of independent assortment of genes. That means during gamete formation there is a crossing over, and during crossing over between this homologous pair, these genes are basically shuffled. So if there is a crossing over between this locus, say locus 1, then the gamete, okay, the gametes will be, they will be separated, and these gametes will be like capital A, then B, C, and small a. This one will be capital B, capital C, right? This will be the different gametes that will be produced from a parental generation. Now, the next question that scientists wanted to answer was, what is the distance between these two loci, or these two genes, or these two characters? So the idea was that if the distance between these two genes are more, then the recombinant frequency will be more. So now I'm coming to another concept called recombination frequency. So what is recombination frequency? And this is very important. If you understand this, then the entire concept will become very easy. So recombination frequency, suppose I have say two genes, capital A, capital B, okay, and then here I have small a, small b, and now I'm crossing it with a recessive individual. And why recessive? Because whatever is the frequency of this particular individual, it will be all expressed because this is recessive. I'll tell you how. So suppose this particular individual, there's a cross parental. So what are the gametes possibilities? The gametes possibilities is that this is N. So these will separate out, okay? So one gamete will be capital A, capital B. The other will be small a, small b. Now the concept becomes a little complex if there is a crossing over. So crossing over capital A, small b, okay? So capital A, small b is like this. And then we have small a, capital B. We have small a, capital B. And that is here. These are the two gametes which are formed as a result of recombination, right? This is formed as a result of recombination. And these two are called parental types without recombination. Parental types. So gametes from two genes, again I'm saying 1 and 2, 1, 2. How many gametes are formed? 1, 2, 3, 4. So from two genes, 4 gametes are formed. What is the formula? The formula is 2 to the power n. 2 means it is a diploid state. That means every copy of gene is in pair. And what is this is in pair, and what is n in number of genes we are concerned about now in a cross. So here we are concerned about 2 genes. So 2 to the power 2 is 4. If we had considered 3 genes, for example, in this case, 3 genes, right? 1, 2, 3. Here, how many gametes possibilities were there? 2 to the power 3, that is 8. Now, let's talk about this 3-point cross. That means 3 genes we are taking into consideration. Okay? Now, why did scientists use a 3-point cross? They used 3-point cross to understand the relative distance between genes. What is relative? Relative means the distance between A and B in relation to between B and C. Suppose this locus is called 1 and this loci is called 2. If the distance between A and B is more than B and C, that is the question that they wanted to answer. That's how they can actually understand how the arrangement of genes are present on the chromosome. Okay? Now, how will you understand? We don't see locus, we don't see genes. At that point, there was no DNA sequencing, no ATGC concept, no DNA structure, nothing was known, right? So, how did the scientists understand whether these two A and B loci are close to one another? Depending upon the recombination frequency. So, this is now coming the important recombination frequency. Okay? So, here recombination frequency. Now, what is that? Recombination frequency is that we can do a cross. Suppose this individual is like this, heterozygous. We did a cross with a recessive homozygous. And what are the gamete possibilities here from this particular parent? We have a non-recombinant capital A, B, C, then small a, B, C. These are non-recombinant types. Now, if there is a crossing over at region 1, this is region 1, we have a crossing over here. Then capital A, small b, small c, capital A, small b, small c. The other homologue will have small a, capital B, capital C. So, it will be small a, capital B, capital C. What will be the case if the crossing over happened only in the region 2? Then capital A, capital B, small c. Okay? Capital A, capital B, small c, and accordingly small a, small b, capital C. Small a, small b, capital C. Now, there could be a possibility when crossing over can happen simultaneously in region 1 and region 2. In that case, it will be a double crossover, crossover 1, crossover 2. So, what was this? This was at crossing over 1, this was crossing over 2. Now, if crossing over happens at 1 and 2, then there will be another 2 possibilities. Okay? So, these are like this. So, capital A, small b, capital C, right? So, let me write capital A, small b, capital C. The other option, small a, capital B, small c. Small a, capital B, small c. Now, these are the gametes. These are the gametes. What will be the F1 if I just overwrite here? I want to overwrite this one. So, if I want to write it F1, then from here, whether there is a crossing over here or both, doesn't matter, because there will be only one type of gamete. That's why we took a recessive homozygous. Okay? And so you can basically write below, if I use a different color for this, say for example, I'm using a blue color. So, capital A, capital B is all small. So, you just can write it below these, and it becomes the genotype of the F1 individual, right? So, it is small a, small b, small c, and I'm not writing it, you understand. So now suppose a scientist go to the field and did a 3-point cross and observes these are the types. Well, how many types? As I said, the formula was 2 to the power n, 2 to the power 3 genes, and it is number of possible 8. See, 1, 2, 3, 4, 5, 6, 7, 8. Okay, so now the scientists go to the field and observe these phenotypes. So these two phenotypes, if he observe, how many individuals? Suppose he counts and writes the individual numbers. And suppose I am writing this numbers like this. This is a real experiment from a real experiment. 1455. Okay. At crossing over region 1, he counts and say find these two, right? So again, writing the actual numbers. Okay. Similarly for these two, he finds, and for these two, he finds only one each. Now from here, what do you take? The take-home message is that the number of individuals he found with this type of phenotype is maximum. So this is non-crossover. And we know why non-crossover is maximum. We know when a crossing over happened between two chromosomes, the chance of crossing over is always 50% maximum. So the non-crossover is always maximum than the cumulative of all crossover. And the crossover 1 and crossover 2 are kind of similar. The case of DCO or double crossover type is DCO, means double crossover here as well as here, is very less because there's a chance factor. One crossing over will prevent the second crossing over less. So from just counting the number of individuals or counting the recombinant frequency, we can actually understand this 4 types: non-crossover type, crossover region 1, crossover region 2, and double crossover. Till now you understand? Now, as I said, what is the aim of this experiment? To experiment is to find the relative distance between A and B, these two locus. So what is the relative distance between A and B? Interesting. It is nothing but the recombination frequency, how many times there is a crossing over between A and B. So here, if you see very clearly, this is the case where crossing over happened. This is the case where crossing over happened. Here, 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 all. So these values are the recombinant types. Okay. So you just sum this up. Okay. And you just sum this up and you will get, so you just sum this up, 163 plus 130 plus 192 plus 148 plus 1 plus 1. Recombination frequency, number of recombinant divided by the total number of individuals. Now you also add these and this. The total number of individuals, it will be how much? 3248. Okay, so it will be actually 295 by 3248. Okay. So, no, I'm sorry. There is a slight mistake here. And it is important to note that the distance between A and B is the recombination between A and B. So, this is a recombination. So, 3 and 4 correct. Not this one, because see, there is no recombination. So, capital and capital B aside to one another. So, we won't take this. So, please, here note that these won't be taken. Okay. These only considered. So, this is 295 divided by the total number 3248, it becomes 9.1 centimorgan. Okay. So again, if you see what is the recombination frequency between B and C, that means how many times this is inverted. Now you will take this one. You see this observation 192, and so if you see 3 and 5 and 6. Capital B small c and small b capital C. Also DCO. So the recombination, so this is between A and B. So what is the recombination frequency between B and C? It is here 192, 148, 11. 192, 148, 11 divided by total number. Total is same 3248. It becomes 10.5. This centimorgan is an unit. Okay, it is actually the value is 0.091. It is, since Morgan discovered it is Morgan, you multiply that with 100, it becomes centimorgan. Similarly, this one also, the actual value is 0.105. Morgan is the hypothetical unit after Morgan, and you multiply with 100 is centimorgan. So now you can actually map the distance. You see that A and B are closer to one another compared to C, because the distance between A and B is 9.1 centimorgan, whereas between B and C it is 10.5 centimorgan. So the initial figure where we started from, like we thought A and B was more than B and C, it's not that. We actually calculated that A and B is closer to one another than B and C. This is called map distance, or mapping of chromosome, mapping of genes on a chromosome. Okay, so now the last part of my talk would be what is the other two concept. The other concept is called the fact it is called interference and coefficient of coincidence. So interference and coefficient of coincidence. What is this? So if you see this too I will cover up in my next video, because it will be too much for one. So interference means, in theoretically, how to calculate I will talk in the next video, but interference means what is the chance of interference of one crossover in the next crossover. It means if there is one crossover, then the second crossover will not take place. Did you understand? So if one crossing over is taking place, then it will interfere with the crossing over in the next region. That is called interference. And so there is a probability. Probability means out of 100. So out of 100 time crossing over happening during gamete formation, how many times that one crossing over will prevent the crossing over in the other region? This is called interference, and that is in probability 100. In 1 minus 100, that is coefficient of coincidence. That means what is the chance of having two crossing over in the adjacent region out of 100. So 1 minus interference is called coefficient of coincidence. Okay, if you find this video helpful, please hit the like button and subscribe to